Hey biology kids, let's go through some of your vocab. So a chromatid is half of a chromosome. A chromosome is when you have two chromatids joined together at the centromere. The centromere is the midpoint. It's the center of a chromosome. When you have two identical chromatids, we call those sister chromatids. It's a double copy of the information that you would get from one parent. A tetrad, then, is when you put together both complete sets of chromosomes, the two chromatids from mom and the two chromatids from dad, to give you a total of 4 chromatids. The diploid number is the number of chromosomes that are present in all of your body cells. That means you have 2 copies of every chromosome, so in humans, that gives us a total of 46. Your haploid number is the number of chromosomes that are present in sex cells. So in humans, sperm and egg cells each have 23, 1 copy of each chromosome. So what I've got shown in black here at the top is one chromatid. The two are joined together to form a chromosome, and in the middle, they are joined at the centromere. And in red, I've got 2 sister chromatids for mom, and in blue, 2 sister chromatids for dad. So if you added up the 4 chromatids, 2 red, 2 blue, that would give you your tetrad. Here I have the phases of meiosis 1 and 2 drawn out. As you can see in interphase, the DNA is loose. It makes a copy of itself. So now in prophase 1, we see the chromosomes appear, and we have a total of 4 chromatids. They begin doing crossover during prophase and metaphase 1, where they meet at the middle. In anaphase 1, they are going to separate, and as you can see, we have a mixture of red and blue on some of those chromatids. Telophase, they're going to divide. Cytokinesis, they're going to split completely into 2 diploid daughter cells. In prophase 2, we've skipped interphase, so there's no DNA duplication. We have 2 mixed chromatids that appear in each chromosome. In metaphase, they line up at the middle. Anaphase, 1 chromatid moves to each opposite end of the cell, and those are going to split into 4 haploid daughter cells. So we've gone from 4 copies of every chromosome to 2 copies of every chromosome down to 1 copy of every chromosome. So the difference between mitosis and meiosis is that we're making body cells in mitosis that are 2 daughter cells that are identical. In meiosis, we're doing crossing over to make 4 sex cells, and those sex cells can vary. Meiosis 2 is necessary to cut the number of chromosomes in half. Crossing over is that mixing of DNA from mom and dad's chromatids, and that occurs in prophase 1 and metaphase 1. Sperm cells, when we create those through meiosis, we're creating 4 equal viable cells. With eggs, we're creating only 1 viable cell and 3 non-functional polar bodies. Here you can read about examples of trisomies and monosomies. Trisomy, we have 3 copies. Monosomy, we have 1 copy of a chromosome. For our learning targets, advantages and disadvantages of sexual reproduction. Advantage is that we have a lot of genetic variation. Species need genetic variation to survive catastrophes. Disadvantage is that you have to go find a partner, and it's a very complex process of meiosis to make those sex cells. For advantages and disadvantages of asexual reproduction, advantage is that it's very simple. You don't need a partner. You also don't need to go through all the steps of meiosis. You just do mitosis. The problem is that you have no genetic variation. All members of that species are going to be identical. So if somebody has a problem, they all have a problem. Meiosis produces non-identical cells because of crossing over that occurs in prophase and metaphase 1. And genetic variation that is caused by this could be the fact that you don't have the exact same hair color as one entire generation of your parents' side. You could have slight variations of that. You're a mixture of both parents. Good luck on tomorrow's test. If you have any questions, stop in first block. I can definitely help you out.